So if you have, so this was very sort of diverse, talks from very, very different corners of the life sciences. And while we were waiting for questions from the audience, I guess maybe I can, so maybe like I can start with questions on ecologies. So yeah, I guess like this is like pretty naive, but so what's the sort of like a downstream process that comes after this like barley? Is this like for some kind of like a food production, or like what's the kind of impact these like microbially produced like substances have to like,
I think the most important one financially is beer production, because it is the main ingredient in most alcohol beverages.
I guess like from someone from sort of Munich, I can sort of relate to.
Exactly. In fact, when I was there, it was very interesting. And when you think about it in that way, the reason why it's important, if you have a lot of microbial contamination, especially at a larger scale, you lose a lot of money. Yes.
But they are, I guess, like sort of contributing to the normal process of fermentation as well, right?
Exactly, they do. But the interesting fact is that in most breweries, they are more interested in the step when fermentation starts. But with this one, it's a process of garbage in, garbage out. If you have terrible seeds that you've stored, it won't even get to the malting process.
It's more of a quality control kind of perspective. If any other speakers have sort of questions each other also.
Maybe I can ask you a question.
Yes.
I just wonder, when you're creating this virtual reality, how do you know that the fish really think it's a reality?
Yeah, so that's a good question. So it's sort of in a way circular. So unfortunately, right now we don't really have like really nice kind of behavioral readout of what these like small animals are thinking. So I guess like I kind of mentioned that one of the strengths of live zebrafish or as a model system in neuroscience is that they're so small, so you can sort of really understand like nitty-gritty details of how they're connected. But like the sort of like flip side of this is that you can't really ask these animals, even compared to like, for example, rodents, it's difficult to sort of like train them to do things. So, like at the end of the day, it's kind of circular, but basically the kind of thing I'm finding, which is like you move around the scenes and you look at the cells, you kind of know already that they are supposed to be compass neurons or like head direction neurons. They move around. This is really sort of the, tells me that, okay, like they are to some extent kind of fooled by this VR. Yeah.
I see.
I have a question. So I have three questions, maybe two for Ms. Hannah and one for Dr. Kalonji. So for Ms. Hannah, I was wondering, because you mentioned about the mechanism of ferroptosis wave. So I was wondering, what is the underlying mechanism, and if actually the cell metabolic activity affects the waves? And maybe the second question is that, because you mentioned lipid ROS, I was wondering if there are other forms of ROS that actually also contribute to the wave, which eventually cause like the cell death.
Sorry, so your first question is, what is the mechanism that is underlying the ferroptosis wave? As we saw in the movie, the ferroptosis propagates at a constant speed. So we think that the mechanism is a reaction diffusion mechanism, such that when one of the cells die, it will diffuse the signaling molecule, which is ROS, to the neighbor cells. And the neighbor cells will undergo this reaction to amplify the ROS again to high levels. And then it will diffuse again to the neighbor cells. So this process iterates in space so that the speed of propagation is constant. And then as to, is there other reactive oxygen species? So right now, we hypothesize that the 4 types of reactive oxygen species are all involved, because by inhibiting one of the 4, the wave will stop. So the four ROS are hydroxyl radical, superoxide, anion, hydrogen peroxide. Yeah. So, but right now we cannot really discriminate like which ROS plays a greater role than the other. Yeah.
By chance, do you look at mitochondria ROS?
Mitochondria ROS. So we also have a scavenger that's specific for mitochondria ROS, like mitotempo, and it can also stop the cell death propagation. So I guess it is involved as well.
Thank you. Just one final question for Dr. Kalonji. So we mentioned like there are different forms of data like metagenomic and metaproteomics. I was wondering if like the proteomic data and the genomic data actually give you the same direction, because sometimes like we do get data that completely points in different direction, like as you know, like from transcription and translation, things just get lost in between and end up we get like two different sets of data. So do you see that, and what's your take on it if we were to get two different sets of data pointing in different directions?
Yes, I think I'll say the metaproteomic was actually the most challenging part of this study for two reasons. I think we've made a lot of progress with the omics data in general, but when it comes to the proteomics data, especially metaproteomic data, which is a relatively new field and evolving, I would say it depends on the database you use at the end of your process to search. I tried two approaches in this study. The first one, as soon as I got my peptide, I searched them against just a general database like the UniProt one, and I got a different set of profile. Then I went back now on the computer, I generated a specific database with my genomic annotation, and I got a different result. And to be honest, I would trust the database that I curated, hence the reason why it's important to follow genomic, functional annotation, structural annotation, then you build the database to search on your proteomics, or else you're going to get anything. Like when you spoke about catalases and all those other proteins are found in 1,000 other species. Yeah.
Please.
Thank you all for the lovely talks. I had a question for Hannah. So you very interestingly mentioned how you're going to leverage this ferroptosis mechanism in cancer therapy, but it also comes with a lot of off-target effects, right? Given that these are ROS diffusing and damaging the nearby tissue.
Yes.
So what are your thoughts on how efficient this method would be in comparison to the currently available therapies against cancer?
Thank you for the question. I think this is really important. I would also like to know all your thoughts about it. I agree that the therapeutic window for inducing ferroptosis can be pretty limited because normal cells as well, they're sensitive to ferroptosis. So right now we are thinking about a synthetic gene circuit approach such that the gene circuit will only be expressed in cancers. So we are thinking about, like, for example, in immunotherapies, there's a cancer-specific promoters that's highly expressed in cancer. So they're the cells that will be targeted. In designing ferroptosis gene therapy, we hope that we can implement similar approach such that the cancer-specific promoter will induce a gene that will cause ferroptosis. So this is what we are thinking about right now.
And are you also thinking of ways to somehow contain how much the damage is happening?
That's really great idea. I think this this might be a little bit more involving, so we need to maybe know, like, if I want the cell death to reach this distance, how much speed I should induce in this cell population. I think it's an important question, and yeah, I think it's an inspiring question that we maybe want to answer very soon.
Right.
Yeah,
Thank you so much.
Please.
Hello. Thank you for the delightful talks. I have a question for Miss Hannah. I just want to ask about cell death, right? So I'm not from the field of biology, so my understanding about this is quite shallow. So this is just a general question. Cancer, in my understanding, is caused by because you want the cells to die, but they can't die because some genes are something went wrong with the genes with the DNA. So I just want to know what is the difference between lipid ROS mediated cell death with natural occurring cell death. What is the difference and what? Yeah, that is the question.
Um, um, yeah. So by naturally occurring cell death, do you mean like apoptosis or
Yes,
the
apoptosis.
programmed type of cell death? So I think I would say, based on our work, that ferroptosis is naturally occurring as well, because it naturally occurs during development of the embryo. So yeah, but so what is the difference between this apoptosis and ferroptosis is mainly the mode of execution. So the signals that will induce apoptosis is different from the signals that can induce ferroptosis. Yes. Does that answer your question? Sorry.
Yeah, so
Yeah.
what
Maybe in the interest of time, we can maybe go to.
Sorry.
Okay, thank
Yeah,
you.
We
sorry.
can talk
Yeah.
later.
I think we can take like 2 quick questions.
Thank you very much for your nice talk. This is, I have a question for Dr. Tanaka.
Yep.
And it's actually a general case of asking for a friend. Yeah, because I have a friend that can claim for the life of her that she cannot tell east from west, north from south. So when I hear that you said that we have molecules in our brain that can help us do that, it's really piqued my curiosity.
Yeah.
So is it because we have that molecule in our brain? Is it more biology limit or a psychology limit?
I guess I'm actually bad at this too. I was super confused around like Merlion, like 2 days ago. Despite I'm saying like you can do this. I think like they say it's a learnable skill. I think like one of the things is that like you can pay attention to things that's far away. That's the only thing that, because like things close by, if you move, the orientation becomes sort of unreliable. So like that's I can say, all I can say. Yep.
Oh, awesome. Good news for her then. Thank you very much.
Hello, I have a quick question for Dr. Chisekedi. So I'm not specialist from the field, but typically in France, regularly we have like barley silos exploding. And basically, to what extent your study about the bacteria within the barley silos can actually lead to a safer silo management, or for instance, identifying at which point of the cycle, based on the bacteria and their respective characteristics, there is a higher explosion risk, or potentially improve or adjust the silo management strategy to avoid actually this risk?
Yes, I think that's where the application of this study will come. If I were to take the study further this level, I think I'll add maybe metabolomics to this part of the research. But at this level, you can start predicting and sorting out the microorganisms capable of releasing such compound. But that's an interesting question. Thank you so much.
Like maybe I can ask one quick question to Lois. So like I come to like sort of life science / neuroscience from a very sort of more psychology kind of. So like maybe I might have missed, but these like long like DNAs, like aside from being biomarkers, like are they like do you have like ideas about like the functions of these things, or is this just Yeah,
I think this is a very good question, and this is actually the question that we have been asking ourselves like every day, because for cell-free DNA, mostly now we want to harness it as a biomarker, but then for its function, it's also an area that is like underexplored, and some has proposed that actually this kind of like cell-free DNA, it is actually it can be some danger associated pattern, and then like it can like stimulate the immune response of the body. Like for example, in in patients who are born with DNA's deficiency, like deficiency in DNase 1L3, they have problem in dealing with the DNA, so they can't digest the DNA well. And these patients actually have familial systemic lupus erythematosus. So it's a generalized syndrome. So actually, cell-free DNA itself, it can be a biomarker. It may cause some diseases, but then in terms of function, whether it plays a role in communication between cells or organs, we might need more studies to elucidate about it.
Thank you. So in the interest of time, we're gonna wrap up here and let's move on to the second session. And thank you for your attention again.
Thank you.
Speaker.